Hello, hello. Janae Carruthers from Science Speak Spokane and Spokane Falls Community College here for you today. We are going to have another lesson on some lab protocols and some exciting components with genetics. But we're going to engage in an activity first. I'm going to ask the moderators to pause, and while we're paused in your Zoom chat, type what you already know about DNA, some vocab words that come up when you think about DNA. Go ahead and put it in the Zoom chat. All right, I hope you engaged with your fellow peers. So when we're talking about DNA, you may have had the word nucleotides, you may have talked about double helix structure. Let's go ahead and start with the human or an animal cell or a human cell. So one of the words you may have said that DNA is found in cells. Remember when we did our DNA extraction with the strawberries, we used a lot of mechanical and chemical abrasion and movement to break up cell membranes and cell walls. So DNA is found within cells. Human DNA, animal DNA, is protected with chromosomes. So DNA is organized into chromosomes. They're also protected by additional proteins called nucleosomes and histones. So those are protection to protect your DNA. Gene is how we label sections or segments of DNA. So genes are just areas on a DNA that produce genetic material. Here's that double helix structure that we're familiar with. So by double helix, it kind of looks like a ladder that swirls. And then if we straighten out our DNA, we have these nucleotides that are labeled by A, G, C, and T. Those are different nucleotides that bind together to make base pairs. So the image that is blowing up on the bottom, those are our nucleotides, and each is a base pair. So A always binds with the nucleotide T, and G always binds with C. So that's sort of our alphabet of what makes us. Nucleotides, again, bind with these base pairs. There's a chemical binding that makes these base pairs. And we're going to be looking at a lab protocol called gel electrophoresis, which separates out genetic material by size and charge. By size, as of today, there is no gel electrophoresis, no lab standard that can look at the entire human genome. The human genome is between 300 and 500 million base pairs. Yes, millions. So we're just going to say 300 million base pairs. Here in the image on the bottom right, that's about roughly 20 base pairs right here. That's a DNA section, DNA fragment of about 20 base pairs. Human genome, like I said, is about 300 million base pairs. How do we examine all of this? How do we look at what the human genome is? How do we make sense of all of that? Humans are roughly 95% made up of the same nucleotide alphabet, roughly 95%. And then we do have some genetic variations that make us unique. So scientists like to examine these variations to focus on genetics, to focus on disease, cancer, and just, DNA is fascinating. It's how it makes us. Another component that makes the human genome so long are these ideas of telomeres. So the image on the left, this is a microscopic image or an scanning electron microscope image of our chromosomes. Remember, the chromosomes are those structures that DNA is organized into. And the little bright lights on the end of each of these chromosomes are what's called telomeres. If you think about your shoelaces at home, a shoelace, and then there's that plastic inlet, that plastic end piece that protects the shoelace from unraveling, that's what a telomere does for our DNA. And this adds to the length of our human genomes. Telomeres are important because every time your cells replicate, you do lose a little bit of your end DNA. And so telomere and telomerase, which is a protein or an enzyme, telomerase, which is an enzyme, helps protect and add this sequence on the back of at the end of your DNA. Telomeres are important because it is a repetitive repetitive alphabet of those nucleotides. And so humans themselves, all humans have this unique repetitive code at the end of all of our chromosomes. It's kind of like I said, like the shoelace. It's a repetitive code of this loose DNA so that your telomerase knows to bind it back, and then you have your completed DNA at the end. This helps, again, as we age, as we replicate our cells due to injury or disease, so we maintain our cells and don't lose our DNA as we're going. So again, this adds to the length of our human genome. Now, let's say that we know a certain gene that's prone to disease, or we want to compare, we're talking about bacteria DNA, bacteria genomes. Bacteria genomes are pretty small, a few hundred to a few thousand base pairs compared to our millions of base pairs. It's this procedure called gel electrophoresis. So this is a lab protocol. Here we have a scientist who is using gel electrophoresis. And what they use is this plastic tray. This is a plastic tray in which a buffer or a liquid is inserted. This buffer is mostly composed of water and then some sort of sugar or electrolyte component. And then a gel, so a square gel electrophoresis, a square gel is inserted. And this gel is similar to Jell-O. It's literally like a pliable gel that's inserted into this buffer. And then at the top here, you'll see the scientist is using a pipette to insert samples. You see the purple liquid, those are samples, using a pipette to insert the samples into wells. Those little square divots are called wells. And each well runs vertically down to show us what is in the sample. So this scientist is inserting one sample of DNA into each of those square divots or a well. And what's going to happen is an electric current is run through the process. So run through the buffer, run through the gel, run through the DNA samples. And the image on the right is the result that happens after a UV light is shown through the resulting gel. So among that purple liquid, among the samples, there is a fluorescent component that's added. And then the result is the scientists can then use UV light to examine where the DNA fragments are. What size of DNA fragments, they're separated by charge and size again. So looking at different samples. So sometimes this is used to compare, let's say, saliva samples of different humans. So comparing saliva samples, sometimes scientists will examine the microbiome, so different bacteria that would exist in a saliva sample. However, when you take one swab with like a cotton swab, and you were to look at that cotton swab, there would be a little bit, a few strands of DNA on that cotton swab. But by itself, that's a really small amount of DNA. So prior to gel electrophoresis, scientists used this another protocol called polymerase chain reaction, or PCR. It's often known as PCR. And what happens is you have that sample of DNA. It could be a sample of soil. It could be a sample of water, of ice, of a human, some sort of sample. And it's put pretty much into this nucleotide soup. And so what happens is you take your sample and you put it in a soup of nucleotides, those A's, those G's, those C's, and those T's, along with a bunch of enzymes and cell components, and also, electrolytes. And what the process happens is you put it through processes of heat and cooling off and heat and cooling off. And whatever DNA is in that sample is what we call amplified or replicated thousands and thousands of times. So again, we took a cheek swab of a human, and let's say we're interested in the bacteria that's living in that cheek swab. The scientist would add certain primers or DNA primers looking for bacteria into this PCR soup along with nucleotides. And any bacterial DNA that's sitting in that sample would be amplified or replicated thousands and thousands of times so we can get a nice big amount of sample, like lots and lots of DNA in one of these wells. Again, so we use pipette to insert that DNA. So one vertical lane, as it's called, one vertical row or lane would have a different sample. So gel electrophoresis is separating out the fragments. PCR is amplifying or replicating the DNA. Now here's a simplified result or a simplified image of a result of gel electrophoresis. This black box, this big black box would be the gel itself. Up at the top, these are the wells or where the sample is located or inserted, where the sample is inserted. Over on the far left, this is what's called a DNA ladder, Now, based on what you're studying, scientists have created standardized ladders that you buy commercially. And you buy a ladder based on what you're trying to find. If you're looking at bacteria DNA, if you're looking at certain types of bacteria, E. coli, M. lupus, if you're looking for certain bacteria, you can buy one type of ladder. If you're looking for human DNA, you buy another type of ladder. If you're looking for skin DNA, you buy another ladder. So there are different standards of ladders. And what happens is as we run the current through, as we run the electric current through, DNA fragments are separated, remember, by size. So if a DNA fragment stops here, this ladder is telling us that DNA fragment is 3,000 base pairs. Remember, those are the pairs of nucleotides. If a DNA fragment stops here, it is a length of 1,500 base pairs. If a DNA fragment stops here, that is 500 base pairs. So based on what we know about base pairs, which one is the longest fragment and which one is the shortest fragment? Shortest fragment is this 500 base pairs, only 500 nucleotides bound together. The longest fragment is the 3,000, so that's the longest. So our samples are in lane 1, lane 2, lane 3, and lane 4. Those are our samples. And the results are the bright up light bars. So based on those samples, sample 1, 2, 3, or 4, which one is the smallest DNA fragment? The smallest DNA fragment is sample 2. It is even smaller than our 500 base pairs. I'm not sure of this exact measurement here, but it is even smaller than 500 base pairs because it went even further. The smallest fragments will it'll go the furthest. Which sample is the largest fragment? Sample 1 is the largest fragment. It is even larger than 3,000 base pairs. Which sample has 2 different fragment lengths? Which sample has 2 different fragment lengths? That would be sample 3. Sample 3 found amounts that are bigger than 1,500 base pairs and just about. So there's 2 different DNA fragments that are just about 1,500 base pairs and a little bit larger than 1,500 base pairs. So the brightness of this band, I'll go back up to our gel electrophoresis. The brightness of the band, the brighter the band, more or less, the more DNA can be found, the more genetic material can be found. So that is why we amplify our DNA with PCR. If we were just to take a sample and run it through a gel electrophoresis by itself, it'd be really hard to visualize. There wouldn't be a lot of DNA to bind to the fluorescent and produce a result. So typically using samples, scientists would use that polymerase chain reaction, that PCR, to amplify so we can get a nice bright band and more reliable research. Also with PCR, you'll have more of your sample, so you can run multiple trials with your sample. Okay, so all those DNA samples, all of these ideas are often produced with a lot of equipment in a lab, but at home, we want to see how gel electrophoresis works. There are ways to test and examine size and charges of items at home. So instead of using DNA and PCR and different temperatures and the buffers, home, we have a component of food dye. So remember, electrophoresis is using a gel matrix to separate molecules by size and charge. So on the left, I just have some food dyes, your generic food dyes that you have at home. And they could be a mixture of different commercial dyes available called red 3, red 40, blue 1, yellow 5. Those are just some commercial dyes that sometimes are mixed together to create one certain food dye. Again, those are industry standards. So we can use gel electrophoresis to examine chemical molecules and separate them by size and charge. So we can use food dye, again, by separating by size and charge. Now, conveniently, food dye and DNA, these are two separate ideas, but both food dye and DNA is negatively charged. That's a chemical fact that we just know. So food dye and DNA are negatively charged. So remember, we're running an electric current through the gel. So on one end, we'll have the negative charge of our electric current. And on the other end of our gel, we'll have a positive end. And electrons, opposites attract in charges. Opposites attract. So electrons are going to move towards the positive side of our well. So both DNA and food dye are negatively charged. So that's how they move through the well. Positive items, anything that's positive, any chemicals or molecules that are positively charged are going to run on the short end of the gel. Any negative molecules or DNA, they're going to move down the long end because they're attracted to their positive terminal or positive charge. So that's how we separate molecules by size and charge. Charged items go towards their opposite attracting to the positive end. And they also separate out by size with smaller fragments moving further through the gel. So again, at home, I don't have a PCR machine at home, but I ran electrophoresis using food dye. So we're going to watch me do my at-home experiment. Please excuse my goofiness, and we're going to have a view of my kitchen science.
All right, I am ready to make our gel for our gel electrophoresis. In our kit, I have some what's called agar agar. Agar agar is a seaweed extract. You can think of it as like Jello. It is a component that you want even distribution of particles. It is semi-pliable, so semi-movable, and it is a component that you heat it up to make it in liquid format. So sort of like Jello, you heat up Jello to make it in liquid format, and then you let it solidify. So you're gonna let it solidify, and then we're gonna run our particles through this gel. And what happens is it's sort of like crawling through a ball pit. Again, if we could make a ball pit as evenly distributed as possible, the smaller particles, the smaller children, will be able to move through the ball pit quicker. So smaller particles will be able to move through the agar particles quicker, whereas larger particles, larger children, are not able to move through as quick. So it separates out the size once we solidify our gel and run it through our electrophoresis. So I have my protocol here. That's what I'm looking at. And it says add 100 milliliters of water to a microwave safe container. So I just happen to have a measuring cup at home. You could use any microwave safe container and measure out, you can measure out cups, or you can measure out milliliters. So I'm going to get 100 milliliters of water. And I just have that in my measuring cup. I'm looking at my measuring cup. There is a particle in here. When you're looking at measuring liquid, you always want to hold it to eye height. And the meniscus, or the water level, should be right at the level of the measurement you want. So you always want to hold it to eye height. I do see there's some particles in here, so I'm going to dump and re-measure. I want to make sure it looks like water. This is what happens when you're in a kitchen. I think I made rice the other day with this, so. Now my hand isn't necessarily steady, so I'm going to set it down and go to eye height and make sure that is at 100 milliliters. I need a little bit more water. Add 2 grams of agar. So
this, I will audio over this. So I'm adding 2 grams of agar. Remember, you can find that in grocery stores. So I'm using my electric scale to measure out 2 grams of agar.
2 grams. 2 grams. Haha, 2 grams on the dot. Okay, put my agar back. So 2 grams agar. Adding my official labeled Tang bag. Thank you, Kat, for labeling my Tang. 0.8 grams.
So Tang is a juice powder. And what we're using Tang for is because it has sugar molecules and it has electrolyte molecules. So we need just a little bit of electrolytes and enough sugar so that when we run a current through the gel, there are ions that can hold a charge. And then you see in my kitchen, excuse my mess, I am going to microwave this agar, water, and Tang mixture. So I'm setting the microwave for 30-second intervals so it doesn't just boil over. And I'm swirling it around. It's good I used a clear microwave safe container because I want to ensure after every interval of microwaving, I'm looking for consistency of the solution. So I'm making sure there are no clumps. I'm looking for consistency, making sure all of the solution and solids are well mixed. You want to ensure, like we were saying in our gel, we want the consistency to be very equal. So I am swirling around, being careful of the heat. Once it looks consistent, I lined a piece of Tupperware with clear wrap and I'm pouring the liquefied gel. And I'm going to leave this at room temperature for about a half hour. What happens then, similar to Jell-O, is the gel cools and solidifies. So it's going to solidify at room temperature after about a half hour. And I'm ensuring that it's more or less even. And now we're going to make our buffer. While we're waiting for that to cool, we're going to make our buffer. So the buffer is going to have the same components as the gel, just with no agar, so it stays a liquid. Remember, the agar solidifies it. So our buffer solution is going to have water, just similar to the gel, and Tang. So it's going to make Tang. So instead of heating it, I'm just going to mix this buffer solution really well. So again, the same electrolytes and sugar molecules, the Tang, as the gel. And we want to, when we're going to run the current, we need conductor molecules, which is the sugars and the electrons, or electrolytes. Now I just have food dye. So just a box of food dye, and then I'm going to use corn syrup. This corn syrup is going to add viscosity or weight to the food dye. As you know, for liquid food dye, it's pretty like watery. Doesn't have a lot of weight or viscosity to it. So I'm going to put a sample of corn syrup with each of the colors. So now we're adding our food dye, and I'm making sure to keep our samples separate so there's no contamination between samples. So our blue food dye, I'm mixing with the corn syrup. And these are the samples. I'm interested in the sizes of the molecules of these food dye, these commercial food dye. So mixing our different colors. Again, mixing the food dye with cornstarch and keeping each separate. And this is similar to how a scientist would do with the DNA in a lab, is they want to keep all their samples separate. And it would be one sample per well. So now our gel has solidified. It's very similar to Jell-O. And I have a Tupperware. So any Tupperware. That is roughly 3 times the width of the gel. So Tupperware that is roughly 3 times the width of the gel. Now I'm going to take aluminum foil. So aluminum foil has metal ions in it. And we're going to put aluminum foil on either end of our now constructed gel box. So since aluminum has metal ions on it, that can be a conductor. We're going to put an electric charge using this aluminum foil. And that's going to become the conductor of the electrons or the electricity that's going to move through the buffer and the gel. So there is one end of our electric conductor. And I'm going to use aluminum foil to make a second end. One end will be positive and one end will be negative. Think, which one am I going to plug in the positive side and which end am I going to plug in the negative side? Which end do we want the dye to run towards? If dye is negatively charged, we're going to have the dye run towards the positive end. That's just how physics works. It runs towards the opposites attract charge. So negatively charged dye will run towards the positive conductor or aluminum foil. So we're making our gel box. And this gel we used is about a 1% gel. There are different percentages of gel that you would use based on the size of molecules or DNA fragments you're looking for. But the standard is a 1% gel, which means 1% agar to liquid or water. And we added that tang again just for a little bit of electrolytes, a little bit of sugars. Okay, so next, I'm going to grab a blunt object, and at home we can use a fettuccine noodle. That is a dry fettuccine noodle. And I'm going to create those wells or those square divots in the well, or in the gel, excuse me. So using the fettuccine noodle again to create a well, square divot. So I'm just kind of poking out or punching out a hole. In the lab, there's what's called combs. So there's standard plastic items to use and punch those wells into the gel. But at home, we can use any blunt object. You can see I'm creating 4 wells because I have 4 samples. In a lab, they have combs from anywhere from 4 to 28 plus. You can create wells because the samples are going to run vertically. So I have my 4 wells. And now we're adding that buffer, which again is a mixture of water and tang, same composure as the gel, just minus the agar. Now it's time to add the batteries. This is going to create an external power source. Remember, batteries have a positive and negative end. So these are 9-volt batteries. And I'm going to insert them together so that we're making an external power source. So positive goes to negative. So making an external battery source. And then this way there will be a positive end exposed and a negative end exposed. Now it's time to load the samples. I'm using another clean fettuccine noodle to take a sample of the blue dye. And I'm going to carefully look, so I'm moving my head back and forth so I can see the well. And just like a scientist would use a pipette and then carefully insert their sample into the well. So I'm taking the blue dye and I'm inserting into the well. You will see some of the blue dye diffusing out into the buffer, the liquid, and that's okay. A little bit of diffusion is normal, but I'm trying my best to insert as much dye as I can into that well and stick it to the fibers of the gel. Now we have our red dye. I took a clean end of that fettuccine noodle, finding my well. And again, inserting the red dye into the well. Again, a little bit of diffusion is happening. That's normal, but I'm trying to make as much dye as possible into the well. And remember, that's why we put corn syrup with the colors, so that there's a little bit of weight with the coloring. Now we have our green dye, another clean fettuccine noodle, just like you would use a clean pipette, and carefully inserting the dye sample into one well. And then we're inserting our yellow dye. And again, moving my head so I can see the well. And I'm inserting a little bit more yellow because I didn't see a lot. Just making sure a lot of the dye sample was inserted in the well. Okay, so now we're going to take our external battery source, and their number is an exposed negative end and positive end, and I use alligator clips and wire. Now, since our food dye is negatively charged, we want them to run the long end of the gel or towards the camera. So I'm inserting the positive end to that conductor or the aluminum foil, and I'm inserting the negative end to the foil closest to the samples. Positive items will run the short end, negative samples will run the long end because they run towards the positive. So here we have the electricity running through the gel box. So there's electrons, which is the electricity moving towards the positive end. So they're moving through the gel, through the buffer, through the samples. And here is our gel box running. And we're going to let that sit for about 45 minutes to an hour and a half. So there's our gel box from home. Now I took some pictures at different intervals. So here's 1 minute. So here is my aluminum foil, again, the negative side. A good sense to tell if my gel box is working is there's some bubbles. Bubbles mean there's electricity running through. And here are my wells with samples of dye. So food dye are sitting in the wells. And again, some diffusion happened. It's a little bit of the blurriness. As you practice, you'll get less diffusion, but a little bit is normal. So there's 1 minute. Here's 5 minutes of running the current. 15 minutes of running the current. 20 minutes, this is 20 minutes. There's those bubbles. Now here are the wells. You can see the wells. And the color, the food dye, staying in its band, but it's moving down the gel. It's running towards the positive side. 30 minutes. Here we have 45 minutes. You can see our color bands. Yellow is a little hard to see, but here's that yellow colored band. We have our blue, we have our red, we have our green. Here's the wells that I loaded originally, so those were the wells. And remember, gel electrophoresis is separating molecules, in this example, food dye, separating molecules by size and charge. So food dye is negatively charged. It ran the length of the gel towards the positive end. And we also separate out by size. So whatever molecules make up these food dyes, they got separated by size. Which two colors have the biggest molecules? Which two food dyes are the biggest? The largest. The largest food dye molecules are the blue and green, because they didn't run as far. The smaller molecules are the yellow and red, because they ran further. They went further down the gel, they're smaller. So this is an example of how you can separate molecules at home, observe, collect hypotheses about charge and size of items. Going back to a DNA sample in a lab, here's another example of what a gel would look like in a DNA lab. So again, the black box is the gel. At the top, you have the wells where you load the samples. This scientist loaded that ladder, or that standard of how you know different lengths of DNA separate, on either side. So the scientist did 2 ladders on either side. And this is a standard ladder for whatever they're studying. So KB is 1,000 base pairs. So this is 3,000 base pairs, KB. 1,000 base pairs, 500 base pairs, and 100 base pairs. So these samples, these DNA samples that the scientists ran, separated out by size. Oops. So the largest DNA fragments were 1,022 base pairs, and the smallest DNA fragments were 765 base pairs. Gel electrophoresis can be used in many, many ways. It can be used to take bacteria samples, again, from soil, from ice, from water, from humans, from animals, and it can separate out so you can identify, oh, I know this, I don't know the exact number, but E. coli has a genome or a specific gene I know about and I'm sequencing for of 765 base pairs. So here there is E. coli present, for example. Again, I'm not sure of the exact base pair of an E. coli gene. I'm making, an assumption or an example. You can also use gel electrophoresis in examining lengths of different genes, different species, comparing species, seeing their genome length or a certain gene. If you pulled out a certain fragment, you knew the length of it. It also can be used in forensics, but it's not a clear answer. So in forensics, if there was a DNA sample, again, at the crime scene, there'd just be a little bit of the DNA. So a scientist would take the sample, amplify it many, many times. So you'll have lots and lots of copies of the DNA using that PCR. And then you could run the DNA sample of what was found at the crime scene with DNA samples of suspects and see if any matches. But again, that's not an exact science. It's only one component of evidence that could happen in forensics. And so there's all sorts of ideas that you can do. So these are DNA gel electrophoresis in a lab. And then we had gel electrophoresis at home. There are multiple ways to do it. So you see this scientist at home used a power pack of 5 9-volt batteries with the negative end and the positive end plugged in. They didn't use aluminum foil. They used, looks like a wire, making sure that the conductor sits on the length of your gel box. And then this scientist had red, blue, and green. Oops. And they're going to run that through their gel. I thank you for your time used today. We again looked at DNA fragments, DNA sizes, and also molecules such as food dye using gel electrophoresis.